So once again, welcome to the 2024 George Corey Memorial Lecture. Next slide, please. Next slide, please. So before we get started, I'd like to say a few words about George Corey. George was a graduate of Princeton University and Harvard Medical Schools. He came to the NIH in 1976 to serve as head of the virus tumor biology section in the Laboratory of Molecular Virology of the NCI. In 1980, he became chief of that laboratory. In 1981, he received the Arthur S. Fleming Award for Outstanding Government Service. And in 1987, the year of his untimely passing at age 43, was elected member of the National Academy of Sciences. Next slide, please. And this is a picture of George with Peter Howley and Malcolm Martin, who's in the audience. Next slide, please. So this slide got a little bit scrambled in the conversion from Mac to PC, but in George's unfortunately short career, he published over 140 papers deciphering the mechanism of eukaryotic transcription, and he used, for much of this work, the SV40 system in very creative and innovative ways. Next slide, please. George had a number of very outstanding trainees and collaborators, some of whom are listed here. Next slide, please. And correspondingly, this lecture series has been given by a number of really outstanding speakers that are listed here. Most recently, Julie Lederman, Julie Pfeiffer, Carla Kierkegaard, Juan Chang, and last year, Britt Glatzinger. Next slide, please. So this brings us to today's seminar speaker, Dr. Susan Weiss. Susan received her bachelor's degree from Brandeis, then did her PhD work with Michael Bratt at Harvard, and then between 1976 and 1980 was a postdoc at UCSF with Harold Barmas and Michael Bishop. Next. In 1980, she was recruited to the University of Pennsylvania, where she quickly rose through the ranks, becoming a fully tenured professor in 1992 in the Perelman School of Medicine. Between 2010 and 2019, she served as Associate Dean for Postdoctoral Research Training and Director of Biomedical Postdoctoral Programs in the School of Medicine at UPenn. Since 2018, she served as Vice Chair of the Department of Microbiology there, and since 2020, co-director of the Penn Center for Research on Coronaviruses and Other Emerging Pathogens. Next. She's received a number of honors and recognitions, far too numerous to list here. Just a few. In 1994, she was elected Fellow of the American Academy of Microbiology, 2008, Fellow of AAAS, and last year, 2023, she was elected member of the National Academy of Sciences. Next, please. Her work focused on pathogenesis of murine and human coronaviruses and Zika virus, interaction of viruses with double-stranded RNA-induced innate immune pathways, and endogenous double-stranded RNA-induced pathogenesis. Next slide. And her talk today is entitled Coronavirus Activation and Antagonism of Interferon Signaling Pathways from MHV to SARS-CoV-2. And if you click one more, we'll see the CME code for today, 50127. So with that, let's please welcome Susan Weiss for the 2024 Corey Memorial Lecture.
Thank you, Eric. I'm really honored to be invited for this lecture. I didn't personally know George Corey, but I certainly do remember him, and I've known some of his trainees that have continued his work over the years. Okay. What is this? How do we switch to okay, there's my title again. Okay, so a brief overview of my talk. Is this okay? The sound okay? Okay, I'm going to give a brief introduction to coronaviruses and interferon signaling pathways. Then I'm going to talk about the role of the 2'5' phosphodiesterase, or PDE, in RNA cell antagonism and in murine coronavirus pathogenesis. That's the model coronavirus MHV. Then I'm going to talk a little bit about MERS and SARS-encoded multiple innate immune antagonists that confer efficient replication, that are required for replication. And Then for probably the second half of the talk is going to be on infection of primary nasal epithelial air-liquid interface cultures that we use as a model for the nasal epithelium with infection with lethal and common coronaviruses, and focused on the role of interferon signaling responses in this model system. And then the sort of bottom line is that interferon signaling differentiates among the human coronaviruses and is really critical for control of human coronavirus infection in the nasal epithelium. So that's my big conclusion. Okay, so coronaviruses, everyone I think has seen this picture by now. It's kind of iconic by now. Coronaviruses are a family within the nidovirus order, named for the nested messenger RNAs generated during infection, with nido meaning nested. The coronavirus particle has sort of gotten famous as well. The genome is a very long 31 kilobase RNA, probably the longest RNA genome, RNA virus genome. It's complex with a basic nucleocapsid protein to form a helical capsid within the virus particle. I don't know if this kind of shows. And the nucleocapsid is surrounded by a membrane, which is derived from the host cell, which is in the blue. And in that membrane, there are 3 proteins. The spike protein that is also gotten a lot of publicity, and this is the protein that mediates attachment to a receptor and mediates viral entry and also cell-to-cell fusion, so it can mediate spread, it can enhance spread. And it's also been the vaccine target for, because it contains major epitopes for the immune response. And then there are 2 other proteins in the viral membrane, the M protein, the membrane protein, and the envelope protein E. These are both transmembrane proteins. They're really important for the structure of the virion and also for other steps during viral replication. And some coronaviruses also encode a hemagglutinin esterase protein. Not true for SARS-CoV-2, but it is true for the human coronavirus OC43. And this is a non-essential protein, kind of homologous to flu HA, where it is an essential protein. Okay, so I want to do a brief history of coronaviruses. Let me see if I can get the laser pointer to work so I can point a little bit. Okay, you can see this better. Yeah, you can see this better now. Okay, so coronaviruses first appear in the literature back in the 60s, and at that point the only human viruses that we knew about were OC43 and 229E. That both caused the common cold. But there were very many animal coronaviruses known at that time. Infectious bronchitis virus, bovine coronavirus, transmissible gastroenteritis virus, and these were important pathogens of animals. So from the late 60s until the first SARS epidemic in 2002, there was a lot of basic biology done on coronaviruses. Nobody really knows a lot about this, but there was a small group of people working on this whole time, and we really learned a lot about how coronaviruses get into cells, how they come out, about the immune response. The furin site was identified probably in the early 80s in the spike protein. And so when SARS, the first SARS, well, here, there's just a list of some of the viruses that were worked on. All this basic biology was done mostly on these animal viruses, very little on the human viruses. So when SARS-CoV, the first one, emerged in southern China in late 2002, everybody was pretty shocked about it. Our field was totally surprised that this could happen, that a human coronavirus could cause disease like this. And it pretty soon became apparent. Oh, and I should mention the first international coronavirus meeting was held in Würzburg, Germany in 1980. There were about 60 people at that meeting, and that was the entire field at the time. But then after the first SARS epidemic, hundreds of people started working on these viruses. And we learned pretty soon after that they had their origin in bats and that they had transmitted to humans through an intermediate species, the civet. The other thing that happened right after that epidemic was that people really looked for more human coronaviruses, and these 2 were identified, HKU1 and NL63, also categorized as sort of common cold coronaviruses, although HKU1 was isolated from an older person with pneumonia, and NL63 can cause croup and bronchiolitis. But they're much more upper respiratory than SARS was. And then things were kind of quiet in the coronavirus world until 2012, when MERS, or Middle East Respiratory Syndrome virus, emerged in Saudi Arabia, causing a smaller epidemic, which still goes on. There's still people being infected by MERS coronavirus. It was much less transmissible among humans, but it's still transmitted as we know now, from camels. So MERS had a bat origin as well, and transmitted to humans through camels, although different from SARS, the camels are a very large reservoir for MERS. So there are camels in both the Middle East and in Africa that have very high levels of seropositivity to MERS coronavirus. Okay, so then things again were pretty quiet until 2019 when, as we all know, SARS-CoV-2 emerged in Wuhan, a different part of China. It most likely has a bat origin. There have been bat viruses identified that are close to SARS-CoV-2, but we don't really know the exact precursor yet. And we don't know if there was an intermediate species. There may be, but that hasn't really been proven yet. And of course, we know that this is the agent of COVID-19. So common coronaviruses are also zoonotic and may have spread to humans as pandemics. So OC43 is believed to have originated in cattle or pigs and transmitted to humans in the late 1890s. In fact, OC43 is quite homologous to bovine coronavirus and also to the mouse hepatitis virus. And it's thought to have been a much more severe infection, and it sort of morphed into a more common cold virus, or evolved, I should say. And then 229E also is believed to have its origin in bats, which transmitted to camels to humans in the late 18th century. And finally, NL63 also is believed to have an origin in bats. So it's really not surprising that these more virulent viruses were transmitted to humans from animals. Okay, so human coronaviruses belong to different genera. So they're either alpha coronaviruses or beta coronaviruses. 229E and NL63 are alpha coronaviruses. Beta coronaviruses, human viruses, fall into three subgenera: the Embeccoviruses, which include OC43 and HKU1, and the mouse hepatitis virus that I'll talk about a little bit. And then the Sarbeccoviruses, which include the two SARS viruses, and the Murine-viruses that include MERS and quite a lot of related bat viruses. So true for both the Sarbecco and Murine-viruses. There are quite a few bat viruses. So all these coronaviruses have very similar genome structure and genome organization, with this very long open reading frame 1A and 1B at the 5' end of the genome, comprising about 2/3 of the genome. And these proteins are translated eventually into 16 non-structural proteins, which I'm not going to talk about very much. And then the spike, E, M, and N genes are always arrayed in the same order in the 3' end of the genome. And what really differentiates among these different genera and subgenera are these so-called accessory genes that encode these small proteins with the numbered by here, like 4A, 4B, et cetera. And these are the proteins that interact or sort of antagonize the host innate responses in general. Some of them are quite well characterized, and I'll talk a little bit about some of them. And that's what really, so you can see that the alpha coronaviruses have a much more simple genome. Just this, this can be 1 or 2 open reading frames, and we see that the beta coronaviruses have a lot more, particularly SARS and MERS have quite a few accessory proteins, and I'll talk about them in a little bit. Okay, so I want to just talk about some very old findings, what we learned from the model Embeccovirus MHV mouse models. So MHV comes in many different strains. They're shown here. Some of them are shown here. And they infect very different organs: the liver, the brain, the lung, the GI tract. The ones that infect animal colonies are generally these GI-focused or GI-tropic viruses. And some of them can infect, can be dual-tropic, like A59 here. So because and it was found very early on, K. Holmes identified the receptor for MHV back in the early 90s, if I remember correctly. And it was found that all these strains use the same receptor, which is CCAM1. So that right away suggests that receptor is not really the only key to pathogenesis or to tropism. So we way back, I think in the 80s and 90s, probably in the 90s, we constructed chimeric viruses, recombinant viruses, exchanging genes between A59 and JHM and sometimes MHV2. And what we found basically by analyzing these genes in animals was that post-entry events were largely determined tropism and pathogenesis. So again, it wasn't all about receptor utilization. And then we also, to look at this a little bit more closely, we wanted to know the role of interferon sequencing or interferon signaling in determining pathogenesis. So we infected interferon receptor knockout mice with some of these strains of virus. And basically what we found was that the mice died very quickly within 2 days, and that virus infected organs that were never infected in wild-type mice. So this JHM that's highly, highly neurotropic only could infect the kidney, the liver, the heart, just about any other organ. So that interferon signaling was really important to restricting organ tropism. Okay, so coronaviruses produce double-stranded RNA in replication transcription complexes, and this double-stranded RNA is part of its natural replication cycle. And this is true for all RNA viruses, and some DNA viruses also produce double-stranded RNA. And this is when sensed by the host, it's a signal to initiate antiviral signaling or innate responses. So coronaviruses produce these double-stranded RNAs in the so-called double membrane vesicles, which are made from rearranged endoplasmic reticulum. These are MERS coronavirus infected human Cal-U3 cells. But despite the fact, so it's thought that the virus replicates in these vesicles to protect the double-stranded RNA from being sensed by the host as this danger signal to initiate antiviral pathways. But despite that, these viruses do induce antiviral signaling. So I'm going to talk just a little bit about the double-stranded RNA-induced antiviral pathways that I'm talking about. So coronaviruses produce double-stranded RNA, shown here, during their replication and transcription. And that's sensed by these different sensors. So MDA5 senses coronavirus RNA. RIG-I does not, for some reason. That MDA5 sensing signals through MAVs to produce interferon, type 1 and type 3 interferon, which then signals through STAT transcription factors to induce expression of interferon-stimulated genes as well as cytokines. But at the same time, these two other sensors sense double-stranded RNA. OASs, oligoadenylate synthetases, there are 3 of them in humans, sense double-stranded RNA and produce 2,5-A, which is a small oligoadenylate that activates ribonuclease L, and ribonuclease L will cleave host and viral single-stranded RNAs, also leading to apoptosis and inflammation. And then the third pathway, double-stranded RNA, is sensed by PKR that autophosphorylates and then phosphorylates EIF2 alpha, which is a translation initiation factor. And once phosphorylated, translation initiation is shut down, also leading to apoptosis and inflammation. And so while these are 3 independent pathways, both OASs and PKR are interferon-stimulated genes, so they are super-induced. These pathways are also induced once interferons are produced. And these pathways were shown to be very important in terms of COVID disease. The GWAS studies early during the pandemic identified susceptibility genes, and among these were type 1 interferon receptor and actually the oligoadenylate synthetases. So viruses in response have evolved proteins or accessory proteins to combat these pathways, these responses. So I want to talk first about MHV accessory protein NS2, which antagonizes specifically the RNA cell activation pathway. And this work was all done with Bob Silverman, who was a very close collaborator for more than 10 years. He recently retired. And so together with Bob, we studied this protein, and we found a very interesting activity for it, I think. It has 2' 5' phosphodiesterase. And here's the RNA cell pathway again. OASs produce 2,5-A, which then activate RNA cell, which then cleaves single-stranded RNA, both viral and host, leading to reduction of replication and also but increase in inflammation and cell death. So NS2 is an enzyme, and it cleaves this inducer of RNA cell and pretty much shuts down this pathway completely. And in the case of MHV, this is a really important antiviral, anti-host antagonist activity. I'll show you why. So using both wild-type MHV A59 strain and a mutant that was unable to, that has an inactive NS2 enzyme. We compared replication in bone marrow-derived macrophages, and we saw this very interesting pattern that wild-type virus replicates really well, up to 10 to the 7 platforming units per mil. That's a log scale on the left, whereas the mutant barely replicates at all. So this NS2 activity was absolutely essential to replicate in macrophages in cell types that have an interferon or host innate response. In fibroblasts, there's really no need for this activity. And then when we do the same comparison in macrophages derived from RNA cell knockout mice, we see that the mutant completely recovers replication. So this activity is really essential for replication for MHV. And we can see here, this is a bioanalyzer assay of the RNA from infected wild-type bone marrow-derived macrophages, and we can see that in the wild-type infected cells, we see intact 28 and 18S ribosomal RNA, whereas in the mutant-infected cells, we can see that there's an activation of RNA cell as evidenced by this degraded or cleaved ribosomal RNA. So because this is a mouse virus, we could actually look, now we can translate it into its natural host and actually look at what happens in the mouse. So taking the same wild-type and mutant viruses, we infect a mouse. We can infect it either intrahepatically and cause hepatitis, or we can also infect it intracranially, in which case we get encephalitis and hepatitis. So again, here's we infect the mouse with the wild-type and mutant virus, and we see really dramatically that we measure virus in the livers 5 days after infection, which is the peak of replication, and we see that the wild-type virus produces about, again, 10 to the 7th platforming units of virus in the liver per gram of liver, and the mutant barely replicates at all. If we do the same infections in knockout mice, RNA cell knockout mice, the mutant totally recovers. So even in vivo, this NS2 is absolutely vital for replication. And the same thing, we just here, this is staining with H&E of liver sections from these mice, and we can see that the wild-type virus causes inflammation in both wild-type or mutant livers, whereas the mutant virus really shows absolutely there's no cytopathic effect or inflammation in the wild-type mouse, but there is in the mutant mouse. And one of the really interesting things we found is when we look at the same kind of infection, but look at the brain, we see that there's equal replication in mutant or wild-type mice with either virus. So that NS2 and the RNA cell activation is really irrelevant in the brain. And I'll tell you in a minute why that's true. So this is a organ-specific innate immune antagonist in MHV. So just to summarize, we worked on this pathway for a very long time, lots of different aspects of it, and I'm just going to summarize a few of the things that we found out. So MHV infection of mice, antagonism of RNA cell, it turns out in myeloid cells, is required for pathogenesis in the liver and not in the brain. And the reason that we think that is that the brain has very low levels of oligodenylate synthetases as well as other interferon-stimulated genes. And it turns out that the RNA cell activation is totally dependent on OAS genes. RNA cell is constitutively, is pretty much expressed similarly and constitutively in all different cell types, but OAS is very variable among cell types and is very low in the brain. And also that RNA cell can be activated independently of MDA5 and MAV signaling in human, mouse, and bat cells. So it really is an independent pathway that can be activated even when interferon can be shut down by various viruses. The phosphodiesterase domains, the PDEs, are also encoded by Murine-viruses, that's MERS and related viruses, and interestingly, some completely unrelated rotaviruses also. And it's believed that it was picked up from the cell at some point, and that the cellular ACAP7 has a phosphodiesterase domain of really unknown function in the host. And then finally, RNA cell activation for many viruses is dependent on oligodenylate synthase 3, not on OAS1 or OAS2 in human and bat cells. And for coronaviruses, there was, for a while, there was a bunch of publications suggesting that OAS1 was really important restriction factor for SARS-CoV-2. and I think it's still not clear what is really the OAS that is important during SARS infections, which is something we're really interested in working on. But it's clearly that for other viruses, OAS3 was the only isoform that really mattered. So from here, I'm going to move on to studying MERS. We initially got into MERS innate immunity because MERS has this homologous PDE to MHV. But then we started looking at MERS more broadly as its ability to antagonize host responses, and then moved on to SARS during their pandemic. So I want to talk just a little bit about MERS and SARS encoded antagonists of double-stranded induced pathways. So here's the MERS genome. As I mentioned before, the MERS genome is more complex than some of the other viruses, and it has all these small open reading frames. So MERS has at least three antagonists. ENDO-U or NSP15 is one of the replicase encoded proteins in ORF1b, and this is conserved among all coronaviruses. And this is a really interesting protein. It's an endonuclease that we believe cleaves genome RNA to prevent the accumulation of double-stranded RNA, so it almost sacrifices itself. But it's really important for the virus to prevent accumulating double-stranded RNA because it's going to induce all these host responses. But MERS also encodes NS4A, a double-stranded RNA binding protein that further shields double-stranded RNA from the host detection. And then NS4B is homologous to the NS2 of MHV, so this is a 2'5' phosphodiesterase, which antagonizes RNA cell. And it also has a nuclear localization signal that prevents the nuclear translocation of NF-kappa B and inflammation downstream of that. So by using recombinant viruses with mutations or deletions in these different proteins, we were able to show that MERS pretty much completely shuts down innate responses via these three proteins. SARS is a little bit different. SARS has an ENDO-U and NSP15 just like MERS does, but it doesn't have a phosphodiesterase or a double-stranded RNA binding protein. And as I'll show you in a minute, it's much less able to shut down innate immunity. It does have all these other small open reading frames, one of which, the ORF6 encoded protein, is believed to prevent nuclear translocation of STATs and really shut down or reduce interferon-stimulated gene expression, although a couple of published studies as well as our own work doesn't show that really happens. So, but there's lots to look at in these other proteins still to be explored. So just to quickly look at how MERS and SARS mutants induce increased interferon signaling, we used lung-derived Cal-U3 cells here. And here's interferon lambda, interferon beta, and one interferon-stimulated gene. And these are qPCR looking at the amount of induction over mock-infected cells. And we can see that here, the SARS in the blue, wild-type SARS, induces very little interferon type 3 or type 1, and a small amount of this interferon-stimulated gene. Whereas the mutant, this is a mutant in the endonuclease, NSP15 induces more of both interferon and these genes. But when we look at MERS, you can see that MERS induces almost undetectable amounts of interferon. But if we mutate either NSP15 or NSP15 plus NS4A, we can see a lot more induction, up to 9,000-fold of interferon during infection of the cell type. And similarly, MERS doesn't induce any of these either PKR or RNA cell, but these double mutants do. So these antagonist proteins are really important to maintain, to keep the host responses down. And if we look at replication, we can see in the case of SARS a very small defect in an NSP15 mutant and larger defects in MERS mutants. But I'll get back to this later on. But so the point is that both of these viruses induce or express interferon antagonist proteins, but MERS much more so than SARS, perhaps correlating with it being much more pathogenic. So just to summarize this little part here, so again, here's the same pathways. We know that the NSP15 or the ENDO-U prevents the accumulation of double-stranded RNA by cleaving genome. And then NS4A, double-stranded RNA binding protein for MERS, will clean up or protect any of the double-stranded RNA that does get produced during infection. And then the MERS also encodes this NS4B protein to specifically shut down this pathway, as does MHV and OC43. So MERS really takes care of everything here. SARS is different in that it does induce some interferon signaling, not a whole lot compared to some of the other viruses I'll show you in a minute. But it does induce these two pathways. But somehow it's kind of amazing. It keeps phosphorylated eIF2 alpha all through its replication, yet it continues to produce its proteins and replicate quite efficiently. Whereas the other viruses really, data I'm not going to show you, they really need to keep this eIF2 alpha unphosphorylated or it's really bad for their replication. And so we don't really understand how SARS works. Despite inducing these pathways, we believe that, or it's been suggested that ORF6 prevents interferon-stimulated gene expression, although we really haven't seen that experimentally. And there are several other small proteins that we're really interested in understanding better. We're working on ORF6 right now, on the ORF6 protein. So at this point, I want to switch to to talk about our most recent studies. All these studies were started during the pandemic. And the big question here was, how does interferon response influence human coronavirus infection in the nasal epithelium? And for these experiments, there's a picture of the nasal epithelium. We used a variety of viruses, a bunch of viruses that infect different parts of the respiratory tract and cause different types of diseases. So MERS, as we said before, causes a lethal pneumonia. It infects mostly the lower respiratory tract, the lungs. SARS-CoV-2, that causes a real range of diseases from asymptomatic to mild upper respiratory disease to lower respiratory to lethal disease in the lung as well. And then NL63 and 229E that both cause primarily upper respiratory disease, and we kind of call them the common cold coronaviruses. And we also use for these experiments human rhinovirus 16, which is a completely different type of virus. And we use it kind of as a control for how a common cold virus will behave. So why do we think the nasal epithelium is important? Well, it's the first line of defense against respiratory pathogens. It's the initial site of viral replication, and it's primarily composed of ciliated cells and goblet cells, shown here, that produce mucus. And it has these tight junctions at the apical side of the cells, shown here, that contribute to the epithelial barrier. The mucus mucociliary clearance mechanism is is an early response to prevent spread to the lower airway. So the cilia kind of push the mucus around, and we believe that that's one of the ways to sort of keep the virus from being aspirated into the lung. And then there's a very high basal level of expression of interferon and other antiviral genes in the nasal epithelium. So the nasal cells may be primed to defend against invading viruses. And there's some clinical studies published where they looked at, by RNA-seq, looking at the basal levels of interferon response genes in the nose, and found that the better levels of these genes was associated with less severe COVID-19 disease. So I think the interferon response in the brain is quite important. I mean, in the nose is quite important. So we use a primary nasal epithelial cell model of cultures on an air-liquid interface to recapitulate the in vivo airway. And all of this work that I'm going to talk about going on now is done in collaboration with Noam Cohen, who is an ENT surgeon at Penn. And Noam has a collection of from hundreds of patients of primary epithelial cells nasal epithelial cells, in a kind of biobank. And so we have a kind of unending supply of cells to work with. And when we're going to use these cells, we put them on transwells, expand them in liquid culture, and then remove the liquid and differentiate them under an air-liquid interface to recapitulate an in vivo airway. And so Here's a little picture, and they look quite a lot like the nasal epithelium. Here's a picture, an EM picture from above. These are the cilia that you can see here. And in this picture, you can see the cilia. You should be able to see the cilia beating. So this is the culture where the mucus has been removed, so you can see what's going on with the cilia. And in this third picture, this culture has not been washed off, so you can see the cilia kind of pushing around the mucus. So after several weeks of differentiation, they're ready to be infected. And so this is the kinetics of replication in these primary nasal cultures, and we can see different patterns here. All these experiments are done with, we now pool cells from about 4 to 6 donors. So, and then we repeat that about 3 times. So by the time we're done, we've probably looked at about somewhere between 12 and 18 donors. This is the average replication kinetics. If we take that apart, we can see that SARS and MERS have kind of similar kinetics. They replicate and then kind of plateau off. These are pretty long cultures. They're going for up to 8 days, 192 hours. If we look at the common cold coronaviruses, they have pretty different kinetics. They replicate early, and then they're pretty much cleared from the cultures. And then finally, if we add human rhinovirus 16, we see that it behaves pretty much like the common cold viruses. So they all have a very similar replication peak and then are cleared. So we also, so there are different temperatures in the upper and lower respiratory tract. With the nose having temperatures in the range of, or the nasal cavity like 32, 33 degrees, and the lungs are more like at 37 degrees body temperature. So we wanted to see how these viruses replicated at the different temperatures, given that they replicate in different parts of the respiratory tract. And what we found was that the cold viruses, 229E, NL63, and HRV16 all showed a similar pattern, which was that they replicated much better at the lower temperature, shown here, 33 degrees, than at 37 degrees. It was pretty dramatic for NL63, for example. So this is the temperature more of the airway, of the nasal airway, and not the lung. But when we looked at the same difference in temperature for SARS and MERS, we saw that for SARS, up till about 144 hours, there really had no preference for either temperature, but then it really crashes at 37, really needs 33 degrees to continue to replicate. And I'll get back to this a little bit later. And MERS really has no preference for temperature. It can replicate equally at 33 or 37. Okay, so the replication of the common cold viruses and HRV16 peaks at early times, followed by clearance. The replication of SARS and MERS plateaus later without evidence of clearance. And just simply the common cold viruses replicate more efficiently at 33 compared to 37. And SARS also replicates optimally at 33 but only at late time points, and MERS replication is not impacted with temperature, by temperature at all. So we wanted to know was how do interferon signaling responses vary among infection with the various viruses, respiratory viruses. We did an RNA-seq experiment, so we infected these cultures with all of these viruses and extracted RNA and then subjected it to RNA-seq. These are volcano plots. I'll put them all up first. Where up-regulation of genes is to the right and down-regulation to the left. And the interferon signaling genes are highlighted in green. And we can see that if we look at MERS, there's very, very little induction of interferon signaling genes. There are a few that are down-regulated. SARS, there's a bit more, as we saw before in the calu cells. But if you look at the cold viruses, it's just way, way more genes induced, interferon signaling genes reduced, and to higher levels as well. And when we quantified the number of interferon response genes induced or upregulated in each infection, we can see that for MERS it's minimal, SARS pretty low, and the other 3 viruses much, much higher. So one of the trademarks of the cold viruses is to induce a lot of interferon signaling. This is just a qPCR validation. I personally trust PCR better. It's really quantitative. So we always do this to make sure that, that the RNA-seq actually makes sense or corresponds with it. And we can see again that there's very little SARS induced by SARS, almost nothing detectable by MERS, and then the other viruses to different extents are inducing more interferon and interferon-stimulated genes. And the other way we look at this, we really like this, is looking at the interferon-stimulated gene protein expression by Western blot. And we can see here that if we look at, this is first for staining for p-stat, phosphorylated stat or activated stat, and just plain stat, which is also an interferon-stimulated gene. We can see, as I said before, that MERS really induces no kind of response at all. Even if we look at any time point, we don't see it. SARS, we see just very late time points, 192 hours. That's 8 days post-infection, nothing until then. Whereas the other viruses, by 48 to 96 hours, are inducing stat phosphorylation. When we look at a bunch of interferon-stimulated genes, we see the same pattern, nothing for MERS, SARS late, and then the other viruses much earlier. And then we also look at PKR activation, just to remind you. We measured this by phosphorylated PKR and phosphorylated EIF2 alpha, and we see again the same pattern, that nothing from MERS, SARS late, and the other viruses at earlier time points are inducing phosphorylation of PKR activation, pathway activation. This is just to show, this is a plotting for the nucleocapsid proteins of each virus, just to show that these viruses are actually replicating to a reasonable extent. So from this we conclude there are like 3 different kinds of patterns, that for MERS there's really no response, for SARS-2 very delayed response, and for NL63-2290 and rhinovirus early and robust responses. So the next thing we wanted to do, we want to know what happens if we take away the interferon signaling response. What will happen to replication of these viruses? So we used a drug called ruxolitinib, and this drug impairs interferon signaling. It works like this. So interferon interacts with its receptor, causing activation of JAK kinase, which then phosphorylates stats, which then can activate or transcribe interferon-stimulated genes. And ruxolitinib inhibits the JAK activity, preventing phosphorylation of STAT and really shutting down or greatly reducing interferon-stimulated gene transcription. So when we treat cells with ruxolitinib or Rux and then infect them, we see this pattern. For the 3 cold viruses that were cleared from infection, they're no longer cleared, and they plateau. They sort of resemble more the kinetics of the virulent viruses of MERS and SARS. When we do the same treatment to SARS and MERS, we see very little. We see a little effect on SARS, latent infection, and really no effect on MERS. So this just sort of substantiates or supports the idea that the interferon response is what's limiting these cold viruses in the nose, whereas these the two virulent viruses really aren't very affected by, or they don't really induce very much of a response. So just to summarize here, human coronaviruses induce a wide range of interferon signaling responses. The common cold viruses induce early and robustly, and the MERS shuts down, and SARS significantly delays interferon responses. The inhibition of interferon signaling with ruxolitinib impairs clearance of the common cold viruses, but minimal effect on replication of the lethal viruses. So we suggest from all this that the extent of interferon signaling in the nose may at least in part determine if infection can progress into the lungs. So if the virus isn't cleared from the nose, it can be more easily aspirated from the lung. So we wanted to know what's the role of the virus-encoded antagonist that I talked about earlier in regulating replication and clearance in this nasal cell model. So these are the same viruses I showed you before, the MERS mutants they're even more attenuated for replication in the primary cell model. And the same for the SARS mutant, which really didn't show much defect in the cell line, is now looking attenuated. But if we and we can look at this again by Western blot, and we see that the SARS mutant induces more p-stat than the wild-type mutant, but both of them do actually induce, whereas with MERS, again, there's no induction by MERS. These mutants induce early and quite robustly compared to the wild-type, and the same thing again for the interferon-stimulated genes. The mutant MERS really induces quite well. The mutant SARS may induce a little bit earlier, but still this delayed induction, whereas MERS is not delayed at all. And if we look at PKR, we see the same pattern, that mutant viruses induce more PKR, not surprisingly, than the wild type. And again, this is just to show that all the viruses are replicating. Okay, so we treated cells with ruxolitinib and then infected with the wild type and mutant viruses, and we see that, interestingly, here's the solid lines are untreated and the dotted lines are treated with ruxolitinib, and we can see that the mutant virus that didn't replicate very well in the presence without the drug now is rescued for replication. So in other words, and the same thing is true with the MERS mutants. So treating or inhibiting the interferon signaling response now allows the mutant viruses to behave as the wild type. So it just shows again that these antagonists prevent the effects of interferon signaling, and once the interferon signaling is gone, they now look like wild-type viruses. So, okay, so in addition to inducing different patterns of signaling, as I showed you earlier, there's also very different patterns of temperature dependence for replication. So we wondered whether does interferon signaling response contribute to the temperature dependence, because we saw these three different patterns both for interferon signaling and also for temperature dependence that sort of along the same lines. So starting with SARS, we asked whether interferon signaling to SARS was more robust at 37 versus 33, and yes, we found that it was. So if we look here, we can see at each temperature there's more p-stat and more interferon-stimulated genes, more PPKR when the experiment is done at 37 degrees compared to 33. However, when we treat these cells with ruxolitinib, we can see that the 37-degree replication is now rescued. So the virus no longer is temperature sensitive when we get rid of interferon signaling. So these two are very, very intertwined, the interferon signaling and the temperature requirement for replication for latent infection for SARS. And the same thing is true with NL63. So NL63 is highly temperature dependent, lower temperature dependent for replication. And if we look at, again, at the interferon signaling response, we see that early in infection, it's much more robust at 37, at 24 and 48 hours. But when we go to the later times, it becomes more robust at 33 because the virus clearly doesn't even replicate out here at 37, replicates so poorly. And again, if we treat the cells with ruxolitinib, it can now replicate at 37 degrees, shown here. Just like the wild-type virus. Just like the 33-degree replication. So inhibition of interferon signaling rescues replication at 37. So just to summarize, so the lethal coronaviruses encode antagonists that prevent induction of interferon signaling and interferon-mediated clearance in these nasal epithelial cells. And inactivation of the interferon antagonist results in phenotypes that mirror the common cold viruses. So we can flip from a virulent virus to a common cold virus with getting rid of these or inactivating the antagonists. And the optimal replication of common cold coronaviruses at 33 is at least part due to temperature-dependent interferon responses. And this was also shown earlier by Ellen Foxman for rhinovirus, similar kind of finding. So there's just one more. This is the last little vignette I'm going to talk about, and that's the question of have SARS coronavirus mutants evolved to be more similar to common cold virus. So it seems from the literature, from observations, that the later variants are more upper respiratory and maybe less virulent. So we wanted to know whether they actually had these characteristics that we've identified of the common cold viruses. So replicating more efficiently at lower temperature, inducing a robust interferon response, and clearance from the nasal epithelial cells. So we initially infected these cultures with wild-type Washington strain of SARS-CoV-2, alpha, beta, delta, and an early Omicron variant. And what we saw was that the Omicron, well, all the variants replicated better than the wild type to some extent, although all the viruses replicate quite well. And we didn't go really far out here. And, but Omicron always replicated a little bit better than the other ones. So Omicron wins the replication race for kinetics. So then we wanted to know what kind of interferon response did it induce, and we again used these RNA-seq in the volcano plots. And here's wild-type SARS. Here's we use delta as a comparison, and here's Omicron. And I've just boxed in the areas that are about the same response level. And we can see that SARS, again, induces a small response. Wild-type SARS, delta, maybe a little more similar, but Omicron really induces a lot more interferon-stimulating response genes. You can see here 14 genes, 23 genes, 51. So Omicron, in this sense, did seem a little bit more like a common cold virus, although maybe not quite to the extent. So then we wanted to look at replication. We thought it would probably have a requirement for low temperature of replication, but we were completely wrong, because here's the kinetics of wild-type SARS, and here's the kinetics of Omicron BA1. It seems to be completely insensitive to the high temperature, so we don't really understand how it does that. But that seemed really sort of quizzical, because how could it survive and spread so well if it has this really strong response? But then we found that it's less sensitive to type 1 and type 3 interferon treatment, and that's shown here. So here's type 1 interferon treatment. That wild-type virus is quite sensitive, whereas Omicron is not. This is by pre-treating cells before infection. And the same thing for interferon lambda or type 3 interferon, much less sensitive. So it's kind of an interesting phenotype that we don't really understand yet. So just in summary of the whole thing, coronaviruses encode conserved non-structural proteins and subgenera specific accessory proteins that antagonize innate immune responses. And in fact, there are a lot more activities within the replicase locus that contribute to interferon antagonism, NSP1, and a whole bunch of capping and methylation enzymes. In the primary nasal cells, common cold coronaviruses like human rhinovirus 16 prefer lower temperature and induce interferon signaling, which leads to viral clearance. Lethal SARS-2 and MERS induce little, if any, interferon signaling and persist in the nasal air-liquid interface cultures and are aspirated into the lung, we believe. Interferon signaling response is more robust at 37, which contributes to the 33-degree temperature optima for the common cold viruses and for SARS late in infection. An Omicron variant has evolved a unique phenotype, which may contribute to its rapid spread. And we're trying to look at some of the more recent Omicron variants to see how they behave with respect to interferon induction and temperature requirements. And then, so finally, induction of interferon signaling in the nasal epithelium can convert a lethal virus into a common cold virus. And we're wondering if interferon is a possible therapeutic approach for these viruses to sort of convert them back and forth with interferon treatment. So just acknowledge the people in my lab. This is my current group of people. Some of these, Clayton Otter did almost all the work I described in the second half of the seminar. He's an amazing MD PhD student who just went back to medical school. And these are Noam's been a really close collaborator. He's right here. Bob Silverman for many, many years. And then these are more recent collaborators, all really great collaborators. And our funding's from NIH. And I'll stop there.
Yeah, please, anyone who has any questions, use the mics, and you want to.
Yeah, questions. Maybe I'll get started. So
Okay.
you mentioned in the introduction that one of the early seasonal coronaviruses that emerged in the 1890s, I think you said,
OC43.
yeah, OC43 was initially highly pathogenic, or maybe thought to be.
Yeah.
Is there anything known about whether the virus changed subsequent to that initial zoonosis, or whether it was the host population that drifted, or is there any remnants of any isolates that are available from those?
I don't know. I mean, it's sort of like wondering whether Omicron becoming more upper respiratory has to do with the vaccine and or the changes in the virus. But I do think that, like, for example, 2290 has they have these two small open reading frames that in the early more clinical isolates were one, and it's an interferon antagonist. So it's certainly plausible that they've sort of lost some of those antagonists. I don't know what the evolutionary drive to do that would be. But yeah,
Maybe not killing the host?
Maybe not kill. Well, certainly that's why Omicron, I think, survives, because it doesn't kill the host. I mean, simplistically, yeah.
Yes.
Thank you. Very, very clean and clear lecture. I was wondering a couple of things. So one, the interferon issue cannot be the sole driver for the temperature differential, right? Because it doesn't work in the case of the last variant. That the interferon responses cannot be the only driver of the temperature differential. And I was wondering if there are other hypotheses that you're working on.
Okay, so well, it could be a major driver of it, I think, because we have looked at like the thermostability of the actual virus particles. We've done that for all these viruses, and they're really all the same. So it's not inherent in the virion. It's not like the virion is unstable. I mean, originally I thought it was going to be the enzymes, like the polymerases or so, maybe more temperature sensitive. But the fact that when we treat with ruxolitinib, when we get rid of interferon signaling, it totally loses that sensitivity, tells me that it's probably a major explanation for it.
That doesn't work for the Omicron, though. For the Omicron, the data that you're showing.
For the Omicron, right. I don't understand Omicron. Take Omicron out of the... Yeah.
So there must be some other issue that's related.
Omicron's not temperature sensitive. It doesn't... Yeah, I don't know. And I have no idea what genes or what proteins in Omicron are giving it that. I couldn't believe that result. It seemed very strange.
And the other question that I had, the culture data clearly demonstrate that when the infection is upper respiratory confined, is self-limiting, essentially, if you want to call it self-limiting, with the contribution of the interferon response. And so in terms of the development of an adaptive immune response, then what's the deal?
So that's one of the things we've been trying to do is, well, we start to put macrophages and dendritic cells or other cell types into these cultures. It's not so easy. But yeah, I mean, this culture can only tell us about the very early steps before there's any adaptive immunity. Yeah. It's a limitation for sure.
You mentioned you use a number of different donors in these primary nasal epithelial cultures. If you look at individual donors, do you see evidence of strong donor dependence, and is there any way potentially to link that back to pathogenesis in the individuals from whom cells were derived? Is there anything to be
I'm really glad you asked that question, because the most striking thing that we found, because in the beginning we did individual donors always, and it turns out when you average them it comes out about the same. But the most striking thing is that there are a lot of donors that seem almost resistant to MERS infection completely. And that's something we're really trying to understand now. We're also looking at similar bronchial cultures from the same person, nasal and bronchial, and how they may differ. But the MERS and the patients or the donors that are so insensitive to MERS seem more susceptible to SARS. And the other thing we did find is if we treat these cultures with IL-13, which is sort of an asthma mimic, or mimics asthma, we get a tilting towards goblet cells, and then, so I didn't mention this, got enough time, but MERS infects goblet cells, SARS infects ciliated cells. So if you can shift that percentage of cells, then you can really see a difference. So it may be that people that have more goblet cells or more, that may be a difference that We're also looking at people with sort of chronic rhinosinusitis and things like that. But yeah, so there definitely is differences among individual donors.
Thanks for a very nice lecture. As I'm sure you know, the Together trial showed that PEG lambda 1 was pretty effective if given early. I just wondered about your thoughts on that and whether you think intranasal lambda or other interferons would be superior or have advantages.
I don't really, I can't, from my own experience, I don't know, but it sounds like something that could be tried. I mean, it does seem to work.
We have a couple questions online. Howard Young up in Frederick. Low-dose oral interferon has been reported to have antiviral activity. Have you ever tried this?
What kind of antiviral? What is that?
A low-dose oral interferon.
Oh, we haven't done that. I think it sounds like it makes sense.
Yeah.
Yeah.
Adam Kaminsky, perhaps I missed this, but in the nasal epithelium behaving in unique ways with coronavirus compared to other viruses, or is it just doing its regular duty?
I think it's doing its regular duty. I mean, the reason we used the rhinovirus was to kind of have a control that would just be an ordinary common cold virus, a completely different type of virus. I mean, one's membrane, one's non-membrane, really different. And so, at least to that extent, I think it's doing its normal job.
Mm-hmm. Anybody else? All right, let's thank Susan for a really beautiful seminar. And there will be a reception right outside now. Thank you. Hopefully it's there.